Bismillah. Assalamu alaikum. Today we will be discussing the complement system, a part of your innate immune system. So, starting with, we'll talk about the antibody structure real quickly because it is important to discuss a bit about the complement system. So, the antibody structure, the points I want to focus on are the FAB region and the FC region. The FAB region is the region of the antibody that is responsible for binding to the antigen. So it is the main part of variability in binding to the antigen. While the FC region is the part that is mostly responsible for the biological properties of the antibody. What we mean by the biological properties is basically the the part of the antibody that determines whether it's an IgM or an IgG or an IgA or an IgD, all of these. So, for example, among the biological properties that are exclusive to the IgM is the ability of the antibody to form pentamers, and that is only and they are only able to do that because their FC region allows them to do it. IgGs, for example, can only make monomers, while IgAs can make monomers and dimers. Also, for example, certain cells have more affinity to bind to certain Fc regions on the antibodies. So some cells will like binding to IgEs more, or IgGs more, or IgM more, and that is what the Fc region is basically responsible for. Now, once we want to start to talk about the complement system, we want to talk first thing about uh, how to talk about it or how to refer to it. And referring to the complement system, we mainly use the plasma proteins that are responsible for the complement system, which are named C1, C2 through C6. All of these proteins, or most of the proteins, will go through a form of cleavage throughout the complement system. And once this cleavage occurs, we will find that an A peptide has been formed and a B peptide has been formed. The A peptide is usually in most of these proteins as the smaller part, and it is mostly responsible for being a chemoattractant. So, for example, we will name it a C1A, a C3A, for example. Next, we have the B peptide, which is usually the larger part, which continues down the complement cascade and the complement system. So we call it a C1b, a C3b, etc. The only exception to this rule is the C2 protein, which is cleaved into a larger C2a that is responsible for the complement cascade, and a smaller C2b, which is responsible to work as a chemoattractant. So C2 is the only exception. Now, once these peptides are cleaved, we will find that they fuse together to form multi-subunit complexes, and they are written as you can see here. This is, for example, the multi-subunit C4b, 2a, 3b. This complex is basically the fusion of a C4b peptide with a C2a peptide with a C3b peptide. These three fuse together to form this one complex. Now let's talk about the functions of the complement system. I want to talk about three functions mainly, and the last function will be basically most of the lecture, while the first two functions will only take one slide each. The first one is the phagocytosis, promotion of phagocytosis, or what we call opsonization. Opsonization is basically, here we have a bacteria. This bacteria, we want the macrophage to phagocytose it. But macrophages aren't always ready to phagocytose. They're not always prepared for it. They're not always activated for it. So in the complement system, the complement peptides, you will find that they bind to to the bacteria itself. So here we find the 4b and the 3b. They are bound to the bacteria. And now here we have a macrophage. This macrophage has complement receptors on it. These complement receptors, once the macrophage comes close contact to the bacteria, the complement receptor will bind to the complement peptide. And once they're bound, it will cause changes in the macrophage to activate it and promote phagocytosis further so it can phagocytose the bacteria effectively. And that's the first function of the complement system. The second function I want to talk about is the recruitment of phagocytes, or what we call chemotaxis. So as I said, the small parts that are cleaved off are all chemoattractants. They all attract basically the phagocytes to come close to them. So here we have all of these, the small parts, 5A, 3A, 4A. All of these are the small parts, small peptides. And here we have a macrophage. And this macrophage, even if it's far away, it will sense these chemoattractants and it will come towards them. And that's what we call chemotaxis due to the complement system. Now, the last function and the one we want to talk about the most, and the one that is most specific and most interesting about the complement system, is the direct killing of microbes. And this is done by formation of a protein or a structure on the bacteria that we call the membrane attack complex. The membrane attack complex is basically a pore. It's just a simple pore in the cell wall and the cell membrane of the bacteria that will allow influx and efflux of fluids and molecules. So once this pore is formed, you will find that the insides of the bacteria are spilling out and the outsides are entering in, and that is incompatible with life, and so the bacteria or the microbe will die by that. So promotion of phagocytosis, recruitment of phagocytes, and the direct killing of microbes are the functions of the complement system. Now let's talk about this direct killing of microbes in further details. So the complement system comes in three different pathways. All of these pathways are activated once infection occurs. So infection occurs, the complement system will be activated. The pathways have different requirements. So for example, the classical pathway requires the presence of antibodies. So not only an infection, but also presence of antibodies in the blood that are capable of binding to this specific microbe. On the other hand, the alternative and lectin pathways, the two other pathways of the complement system, do not require antibodies and can occur in the absence of antibodies. So in case we have an infection where there is antibodies, you will find that all three pathways are occurring. While if there is no antibody, you'll find only the alternative and the lectin pathways to be occurring. All these pathways start in different ways. However, they all converge at one point where all of them form a protease we call C3 convertase. So this C3 convertase is basically where all of them will convert together. And the C3 convertase, from its name, it will cleave C3, resulting in a C3b and C3a. This C3b is extremely important in the formation of C5 convertase, which is essential for the formation of the membrane attack complex. So once C3b is formed, C5 convertase can be formed, and the membrane attack complex can be formed. On the other hand, also, C3b can bind on the phagocytes and facilitate phagocytosis, basically, as we discussed earlier. Now let's talk about each one of the pathways in detail, starting from the classical pathway. So as we said, classical pathway requires the presence of antibodies, either IgD or IgM. It cannot work on IgA, IgD, or IgE, only the IgG and IgM antibodies. These antibodies will bind to the bacteria. Then the C1q peptide will go and bind to the Fc region of the antibodies. And this will cause changes in the peptide, exposing some sites for binding, basically. And these sites will bind to two molecules of C1R, and these C1R will bind again to two molecules of C1S. And thus we form the C1QRS complex. The C1QRS complex is capable of cleaving a few of the complement peptides. Specifically, we're talking about C2 and C4. So C2 and C4 will, over here, they will be cleaved into the large A for C2 and the large B for C4, and the smaller parts will go and through the bloodstream and work as chemoattractants, basically, while the large parts will bind together. Once they are bound together, we get our first C3 convertase, which is C4b2a. This is a C3 convertase in the context of the classical pathway. Of course, as we said, they all converge at this point of formation of C3 convertase, which will cleave a C3 peptide into a C3b and a C3a. And once that is done, the C3A, of course, will go into the bloodstream and work as normal, while C3B will bind to the C3 convertase, forming C5 convertase, which is in the context of the classical pathway C4B, 2A, 3B. Okay, and now C5 convertase will basically cleave a C5 molecule into C5b and C5a. The C5a again will work as a chemoattractant, but here the C5b will go and bind to the bacteria itself. And it will continue through the cascade of complement, but we will stop here because all the pathways will reach this point at one point or another. And so we'll stop here, we'll talk about the other pathways, and then we will see how does this develop. So we have now the lectin pathway. The lectin pathway is extremely similar to the classical pathway, except the beginning. So the beginning, because we said it does not need antibodies, but it needs carbohydrates on the bacterial cell wall or the cell membrane. Specifically, it needs the mannose on these, specifically mannose on the wall. Here we have the lectin, and we call it a mannose binding lectin because, again, it's a lectin that binds to mannose. And this lectin is surprisingly structurally similar to the C1QRS that was formed in the classical pathway. So the lectin will work the same functions that the C1QRS did, and so it will be the same pathway where the C3 convertase is made, and then C3 is cleaved to form a C5 convertase, and that will cleave the C5, and the C5b will bind to the cell wall and the cell membrane of the microbe. And that's the lectin pathway. Again, it's exactly the same as the classical pathway, except that it's initiated by lectin binding to the mannose on the surface of microbes, while the classical pathway is initiated by the formation of C1QRS by binding to antibodies on the surface of the microbe. Now for our last pathway, which is the alternative pathway. This is another pathway that does not require antibodies, and this pathway starts by the cleavage of C3. This cleavage can occur due to different enzymes in the bloodstream or by the um, microbe itself. It can cleave the C3. So it's cleaved into C3b and C3a. Of course, C3a works as a chemoattractant, while C3b will bind to the microbe. Once C3b is bound to the microbe, you find that there is a factor called factor D and a factor called factor B also present in the blood vessel. The factor D will work on factor B and destroy it, or like cleave it, forming BB and BA. The BA is the smaller part, which will work as a chemoattractant, while BB will bind to C3B, forming the complex C3B-BB, which is a C3 convertase, but it is an unstable C3 convertase. How do we make it stable? Simply by binding another factor P. So factor P binds to this complex, forming the C3b-BB-P, which is a stabilized C3 convertase. And now that we got our C3 convertase, again, the pathway will basically converge to cleaving a C3 into C3b and C3a. C3a works as a chemoattractant. C3b will bind to the C3 convertase, forming C5 convertase, which will cleave a C5 into C5 B and C5A. The C5A will work as a chemoattractant, while C5B will bind again to the cell wall of the microbe. And now we will talk about what happens next. So we're done with the three pathways. We saw how each one of them works, and we have a good visual form of how they work. Now let's talk about the C5b. So here, simply what happens is that the C5b, once it's inserted into the membrane, it will facilitate the insertion of C6. C6 will facilitate the insertion of C7. C7 facilitates the insertion of C8, and C8 facilitates the insertion of multiple copies of C9. Once these multiple copies are inserted, they're inserted and they form a pore together. This pore is what we call the membrane attack complex, and it will allow influx and efflux of fluids and molecules through it. And here, as we can see here, this is a microbe and it has multiple MACs on it, each one of them allowing influx and efflux of different fluids and molecules through the cell membrane, which is incompatible with life, and it will result in the death of the microbe. This image has been taken from the Lippincott's Immunology textbook. Now, lastly, let's just discuss the regulation of the complement system. So regulation happens by four proteins mainly. One of them is the C1 inhibitor. C1 inhibitor, from the name, is extremely obvious what it does. It inhibits the C1. C1 is prominent and present in which pathway? The classical pathway. So the classical pathway is inhibited by C1 inhibitor. Another factor is decay acceleration factor, DAF. DAF will block the formation of C3 convertase. How? By stopping the binding of BB to C3b. Where did we see this? We saw this in the alternative pathway. And so DAF is only responsible for inhibiting the alternative pathway, not the classical pathway, not the lectin pathway. So it stops the formation of C3 convertase by stopping the binding of BP to C3b in the alternative pathway. Next, we have factor I. Factor I simply destroys C3b. C3b is extremely crucial in all pathways, in the classical, in the lectin, and in the alternative pathway. And so a factor that destroys C3b will inhibit all of these pathways. Lastly, we have complement receptor 1, which is capable of displacing C3b from its place. So C3b has to bind to some places. The complement receptor 1 will be able to displace the C3b from its place, and that's why it will promote the degradation of C3b. But it also works as a cofactor for factor I, and as we said, factor I destroys C3b. So not only does it promote its degradation by displacing it, it also promotes its degradation by activating the factor that is responsible for its destruction. And that is basically the video. Thank you for listening, and here are my resources. Thank you very much and have a good day.